Good morning, students. Can you all hear me better?
Yes, Professor.
Thank you.
Good morning.
Morning.
Good
Good
everyone.
morning.
Good morning. How is everyone doing?
Great.
Good.
Great. Nice bright
I
sunshine.
have death in my family.
Oh, I'm so sorry to hear. Accept my
Yeah.
sympathies and my my sincere condolences, Reiko.
Yeah, I might not be there tomorrow.
Okay. Thank you for informing me.
And Michael said he's at jury selection, so he might not be here today,
Okay.
but he's
Great.
gonna try to log in.
Okay. Thank you for informing me. Reiko. Just get the notes from someone, and always you can come on office hours, Reiko, just to catch up on what you don't understand.
I did all my graphs, but I'll speak to you after.
Sure.
Okay.
Sure. Okay. Okay. Maybe we just give 1-2 minutes. Yes. Someone wants to say something? No? We'll just give 1-2 minutes for students to get in. Yeah. Okay. And then I'll take attendance. So, I'll get everyone. So tomorrow, y'all know what's tomorrow's lab after cell respiration? What is the lab for tomorrow, students?
Cellular something.
Cellular division.
No, yes, Joshua. No, Arlene, it's cell respiration we did last week. You
No, I said cellular something.
cellular division, correct. Cell
Yes.
Yes. And actually, you will know because we've already been talking about cell division last week in the lecture, and I'm almost done with cell division chapter. So when you come into the lab, you're going to be looking at slides, and you'll be making your own onion root squash of the onion root tip squash. As I mentioned last week when I started with cell division, okay? Okay, maybe 2 more minutes. I'll wait 5 after 11 and then we begin. So some students are logging in. And then I'll take attendance and then we will, I'll continue with the topic on cell division and then we'll do, which is the first one, mitosis, and then we go into the second cell division, which is meiosis, okay?
Professor, I thought you said today was a test.
Today's March 29. Your exam is your second exam. Today's 29th, right? March? No, your second exam is in April.
April 5th.
The 5th. Yep.
April 5th. Sarata, look at your announcement. All the dates of the exams with the chapters are there. It's April 5th. Okay. Next week, Sarata, you have still 1 more week to prepare. That's good news, right, Sarata? Joshua, were you saying something? Did I hear your voice, Joshua?
Oh, I was just saying that the test is on the 5th.
Yes, the second exam is on 5th, correct. I'll just give 1 more minute, 11:05, 5 after 11, and we're going to begin, okay? Yes, I encourage you students to go through those review questions that I put up. And of course, emphasize, remember I said to you on the cell membrane structure diagram, you're also going to emphasize on the plant animal cell, right? What are the different organelles? As you're studying the organelles, keep your PowerPoint slides, look at your textbook also, and also go through the handout notes, that organelle Word document notes, which I had also attached in my folder. Look through everything and always ask yourself questions like the way I formatted my lecture. So that would enable you to remember your information, okay? And always look at a diagram, study with diagrams, it's easier. Okay, it's 11:05. I'm going to begin. Once again, I say good morning to every student in this room. Naba is here. Naba?
Yes, present.
Okay. Alan? Alan? No. Brandon?
Here.
Brandon? Okay. Emily? Emily? No. Amy? Amy? No, this person's not here. Okay. Mumtahina?
Here.
Okay. Natasha?
Present.
Okay. Errol? 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 Not here. Not yet. Fatima? Fatima?
I'm right here. Good morning.
Good morning. Ada? No, not this person. No. Arlene is here. Okay. Naomi? Naomi?
Here. Good morning.
Good morning. Pamela? Pamela? No. Fanila? Fanila?
Hello, Professor.
Hello. Michael, yeah. Okay, so Michael may be on jury duty. Okay, just like that. Reiko is here. Tanjima? Tanjima? Tanjima?
Here, Professor.
Hello. Yes. Andrea? Andrea Michelle? Andrea Michelle? Andrea? No. Angel? Angel? Angel?
I'm here.
Okay. Ali? Ali? Alessandra?
Here.
Okay.
Hi, Professor. Good morning.
Good morning. Sarata is here. She answered me. Joshua answered me. And Brittany? Brittany? Brittany Cyrus? No. Okay. Anyone else's name I didn't call out? Who's on the, everyone's name I called out. Okay. So I'm going to now open, everything is opened here. And I'm going to share my screen. So students, just to let you know where we stopped, okay? So we had come to the slide on the cell control cycle. That's where I had written, I stopped and I was continuing on slide number 22, cell cycle control, but we talked about this cell cycle control system. This is the slide. So I'm going to open it out here and explain the slide from here. Okay, what I want you to do, students, is also go through this. Remember, through the stages or the phases of mitosis. That's what we're going to do in the lab tomorrow. And you are going to look at a permanent slide first on the onion root tip. I'll show you exactly in the lab where you're going to focus on the onion root tip to study all the phases of mitosis. And also to study interphase, and we are focusing on interphase when we talk about the cell cycle. That is before the cell is beginning cell division. What I need you to know is, if you don't recall, but at least have that Word document notes on the cell division that I've put up, on the stages or the phases of mitosis, that is going to be the best reference for you to identify these stages under the microscope tomorrow. So I encourage you to bring that handout on the notes that I gave you, because those notes, as I told you, is a compilation of the exact stages, the exact points, or what I like to say, criteria for helping you to identify all the stages of mitosis as well as interphase under the microscope. Okay, so bring that tomorrow so it will be easier for you. And you're going to spend time first on the permanent slide and then on making your own squash. Isn't that going to be lovely? You're making your own sample and looking at the stages. Now, if you work well on the sample, on the squash, you will be able to see all the stages, including interphase, which is not part of mitosis. Okay, so go through this because this is very important for you to identify. Okay, so now when we are talking about this control system, right? So this is I mentioned to you earlier that this is the cell cycle. The cell cycle now has the starting point. Remember the interphase, which was 90%, and which included G1S, G2 phases, and then the division phase. So all that was the growth, and then the division was mitosis. Now we're asking ourselves, okay, this cycle is going on. All the cells in our body are undergoing cell division. But remember, for the cells to, I'm going back to the initial lecture. For all our cells to undergo division, they must grow. And that growth is taking place in the interphase stage, which is all this G1S, G2. But now we need certain factors. We need certain proteins to control the cell cycle, to control how our cells are going to grow. So we have certain checkpoints, and that is called the cell cycle control system. From the very word control system, it controls the cells to undergo the cycle. But how is it controlling the cells to undergo the cycle? It has proteins. And we're going to learn these two names, CDKs and CDK, cyclin-dependent kinase. You know, proteins are enzymes. So it requires that enzyme too, along with the proteins, cyclin-dependent protein. So give me 1 minute, excuse me. And so now we need to know that there are certain checkpoints. And you know, the classic analogy I use is when we're coming from a different country back into the US, we go through immigration checkpoint. That's our checkpoint. And immigration is checking for our statuses, right? Same thing in our cycle. We have checkpoints like gates. Think about even when you enter BMCC. We have an ID card. We just have to tap the ID card, the turnstile opens. If your ID card is not, identification card is not programmed, it won't, the turnstile doesn't open, the gate doesn't open, and you can't enter BMCC, right? Or any of the buildings, Murray or Fitterman. So the same analogy. So we have now, where are our checkpoints? We have only two checkpoints. We have one checkpoint called G1, so it's at the G1 phase, remember, and one at the M phase. So after G2. So now what are these checkpoints checking for? These checkpoints are checking to see. This is not given in your slide. In one of my non-science major textbooks, it's a beautiful slide. They tell you exactly. They're checking for three things. And if you want, please write this down. They're checking to see if the size of the cell is proper, the nutrient supply is adequate, cell size is proper, nutrient supply is adequate, and cell growth has taken place. Three things they're checking for. Okay. So now, if by chance one of these three factors are not proper, the cell, this is key in understanding now, the cell has to exit the cell cycle and it will go into a phase which we call G0. Now here they don't mention this G0 phase. So now they sit in that phase. G0 means it's a resting stage. But resting means it's not that it's not doing anything. It has to complete what it's missing. You know, some of the cells, they will sit in G0 for days, for weeks, for years. And they can, one of these criteria which they're missing may not be met with, then they die. They can't re-enter the cycle. But if they fulfill any of these criteria, then the gate opens and they can come in. Same thing. So here they're checking for the growth, whatever happened in G1 phase, synthesis, DNA, RNA, protein synthesis. Then they come to G2. G2 also, they're seeing whether there is precision of all the DNA, that the DNA is precisely formed. DNA replication occurred. Size is proper, nutrient supply. When I say growth has occurred, we are saying DNA replication has taken place there. Then the gate opens and now interphase is over. So now the cell has its full complement of genes. It's ready to undergo cell division, namely mitosis. So all this G1S, G2, which was interphase, was the growth phase. Remember in the beginning, I told you cell cycle consists of an alternating sequence of growth and division. So growth has taken place and now the cells are ready to undergo division. Think about it, students. I keep emphatically stating this. It's very important to understand that if growth has taken place, duplication of chromosomes, DNA synthesis, RNA synthesis, protein synthesis takes place, all is not completed here. And then the DNA that is being replicated is replicated with precision. Then what are we saying? There aren't 46 chromosomes in the cell. So how will the cell now begin to divide? Yes, the cell can divide. But what is, the growth has taken place. But when the cell is undergoing this M phase, which is mitosis, the division phase, that means the cell doesn't have its full 46 chromosomes. So the cell is abnormal. And then when it is going through the division, what type of cells are being produced? All abnormal cells with half the genetic information or incomplete genetic information. So it's important to understand the concept. Why should the growth, the interphase stage, be correct? What is happening? What helps the G1S, G2 phases to take place? Any questions, students? Are you all understanding? Any questions? So now we have come to, since we understand the whole concept of the cell cycle and the control, we're going to see now that the cell cycle clock. I am sharing my screen, right, students? Students?
Yes, Professor.
Yes.
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 Okay. Because I keep explaining and then I'm not sure. But I remember saying share. Okay. So now remember I told you that there are two, proteins are enzymes, the enzyme. You know immediately when it's an enzyme, the suffix is aces. So how is the cell cycle clock being controlled? By cyclins and cyclin-dependent kinases, CDKs, K apostrophe S, CDKs, we call them, okay? So what are these cyclins and the CDKs? They are regulatory proteins that are involved in the cell cycle control. See how we write CDKs, lowercase DKs. Now the activity of a CDK rise and falls with changes in concentration of its cyclin partner. We have something called MPF. MPF stands for maturation factor, promoting factor. What is the MPF? It's a cyclin hyphen CDK complex. So when cyclin goes and attaches itself with the cyclin dependent kinases, it forms a complex called cyclin CDK complex. That is the maturation promoting factor. Why is that essential in the cell cycle? Because it triggers a cell's passage. It triggers the cell to go past the G2 phase checkpoint and now enter cell division, which is mitosis. So you could be asked a question on this. These are important slides. Okay, mark these slides important. So let's go now here. I want you all to look first. Let's go to each graph. Remember, I keep asking you to interpret a graph. That's why I give you graphs in the labs. Can you read a graph? Can you interpret the graph? Can you analyze the graph? Can you conclude what the graph is? So I'm teaching you to keep reading graphs and to interpret them correctly. So I want you to take time here to look at A. A says fluctuation of MPF, which is maturation promoting factor activity, and cyclin concentration during the cell cycle. Take 2 minutes, and I would like one student to explain what they think is happening. Okay. Yes, students, would someone like to try? What you understood from this? What do you understand from the graph here? Try someone.
MPF, Professor, MPF is rising up and down.
Okay,
When it reach up a certain part, it's cycling.
1 minute, just go little, when it reaches what?
When it reach its
You're
second,
talking about this graph, no? Which I'm pointing.
yes,
Good.
yes. So when it go to the second curve up, cycling come in.
This one?
Yes. It has
So the-
cycling is happening there. Then it go down again, and then it went back up to normal MPF. So every up and down, it's cycling.
Okay, good. Now I want, if you can be a little more specific when you say up and down. Look carefully.
So
Okay.
it starts on the M. So when it go to the next M, it's cycle. Then it come down and go back to the next M. It's on the M and F. Then it go back down and it go back up again to the next. So every full cycle, every other cycle, it's cycling.
Okay, you're on the right track. Good. Anyone else wants to supplement, add to that explanation? Anyone else would want to try?
Yeah.
Ali?
The way that I see it,
Sure.
so going off of the metaphor that you had said earlier with like the immigration process, I would say cycling is like the agents, right, of that process, and they kind of like push you forward into the next step to get to where you're going. So when we approach the reason why the M phase is happening is because of the increase in cyclin concentration. And the graph on the right, I guess they combine together.
Ali, just talk about this one, then I'll give you time for this one, okay? The B one. Are
Yeah,
you referring to the B graph or only this one here?
the A1. I was talking
Okay,
about the
okay.
yeah.
So are you referring to this last one here?
Yeah, correct.
Okay.
Yep. And yeah, that's why we're seeing the rise and fall of this cyclin activity to
Okay.
compare to the M phase that's being introduced into the next growth phase when there's no more cyclin.
Okay, good, both of you on the right track. So what I would say is that this maturation promoting factor activity, right? What is the relationship? Why should maturation promoting factor activity, which is nothing else but your cyclin and the CDK, where is the concentration the highest? The concentration is the highest when it reaches the M phase when mitosis occurs, which is the division phase, okay? And so it reaches its peak in M phase. And then what you see happening to cyclin concentration, again, cyclin concentration is slowly going to increase, reach its peak again at mitotic stages, and then it's going to drop. And it is going to, no one spoke to me about this part here, which is important. What happens here? It's going to stabilize, reaches equilibrium in the interphase. See, G1, you have to look at all these phases on top as well as match it, okay? So what we are seeing here is it reaches equilibrium. So it increases. When cyclin increases, the concentration of cyclin activity, cyclin concentration increases. When the cell is undergoing mitosis, it reaches its peak at cell division, and then it slowly drops the MPF activity, and then it stabilizes during the, look at this whole part, it stabilizes in interphase. And then again, because remember, interphase is is where the cell is growing, and then it has to grow, get everything that I just spoke about, what it requires to undergo cell division. And then at the M phase again, the cyclin concentration is going to increase, and then it slowly drops during G1S, G2. But it's important to see that in the interphase stage, that it will drop the concentration and then it stabilizes, then again it increases, the concentration will increase. Now, what do you think is happening in the B1? What is the molecular mechanism that is helping to regulate the cell cycle? So I want you to look at this. This is important. I want you all to understand this. What is happening here? Look carefully at the phases, okay? Excuse me. Would someone like to try? Or you are still trying to figure that out?
I'll go for it, Professor.
What's that?
I'll go for it.
You confirm?
Are you, see, I'm totally not even here. Oh my God, I thought you asked somebody to try to explain it and I'm so sorry.
Yes, try, Reiko. Try. You would like to try? Reiko?
Yeah,
Try.
okay, yeah. Let me try.
Sure.
Okay, so as it goes into the GI stage, I see that
G1.
some of the cells have not, G1, I'm sorry.
Correct.
That some of this did not match up. They wasn't incomplete. They wasn't completely, and they need more repair. They're not proper cells. They're not proper cells, so they had to be discarded. And as it goes around the CDK, and then they go to the second checkpoint, which is the G2, and the G2 allows it to have permission to go into the MPF so that the activity can start to fluctuate, and then it could start the whole cycle again and again and build in new cells and bring into new things. I think that's where we are. And I'm so sorry, class, if I'm not making any sense, but I'm trying.
No, very good try, Reiko. That's the whole general concept, good try. But over here, we want to just see a little more what they are indicating here about cyclin getting degraded. Why is cyclin being degraded? When is it degraded? When is more cyclin accumulating? Very good try, Reiko. Would someone else want to try with the explanation? Good attempt, Reiko. Would someone else like to try? See what is happening here, students. You see CDK. What is CDK? Cyclin-dependent kinases, right? What is happening to the cyclin-dependent kinases here? Remember, it's the enzyme. So what does CDK do? CDK is going to break down cyclin. So as it breaks down cyclin, more of the cyclin is going to start accumulating because the enzyme is breaking down the cyclin. More accumulation of cyclin is taking place in the G1-SG2. Once it reaches the G2 checkpoint, what happens again? The maturation promoting factor, which is the C cyclin and the cyclin-dependent kinases complex, is going to act, break down more cyclin, and all that cyclin that is being degraded is now eliminated. That's how the cell cycle is taking place, okay? And more cyclin is going to be degraded where? When it reaches in the mitotic phase, in the division phase. So we are seeing here, I've talked about all this, right? G1S, G2, and this is what they're telling you about the G0, which is the resting stage or the non-dividing state that I called, where the cell is going to exit the cell cycle and prepares for whatever it requires that it's missing, and then come back for mitosis, okay? Now, besides the CDK, Cs, which is cyclin and the cyclin-dependent kinases, which are the MPF, we also have external factors. External factors are going to influence the cell division. And what are those external factors? They will include specific growth factors. From the very term, students, growth factors, that means they're enabling the cells to grow. So growth factors, they are released by certain cells and they will stimulate other cells to divide. Now, what is one type of a growth factor called PDGF? You must know the acronyms because in the exam, you may not get the name of PDGF. So you must know what PDGF stands for. It stands for platelet-derived growth factor, GF growth factor. Now this platelet, we've heard of blood platelets. Our blood consists of red blood cells, white blood cell, blood plasma, and blood platelets. So what are platelets? This PDGF is made by blood cell fragments. Fragments of blood cells are referred to as platelets. And the PDGF is being made by these platelets. Now we have something called density dependent inhibition. To inhibit is to stop or prevent. Now, in density dependent inhibition, the crowded cells will stop dividing. And we're going to see that, okay? So let's look at this figure here. So we're seeing here, this is what we do in cell tissue culture. We take a sample of tissue and we know chapter 1 characteristics, hierarchical organization, cells form tissues. What are tissues? A group of cells. So you take a tissue, a sample of human connective tissue is taken, and this is the scalpel, which is the blade, and it is cut into small little pieces. This is the petri dish, which contains nutrient agar or agar, which is the nutrient medium, not for the growth of bacteria, but for our human cells to grow. Okay. Now what happens here? Remember, we have used Erlenmeyer conical flask in our experiments when you took enzymes, etc., for the enzyme degradation lab. So this is a conical flask called an Erlenmeyer flask. So we add enzymes to the solution. And what happens, you can see the cells here. The enzymes are going to digest all the extracellular matrix, resulting in a suspension of fibroblast cells. So fibroblast cells are going to grow in that medium, in this proper tissue culture medium. Now, from this Erlenmeyer flask, we use certain culture vessels. And these culture vessels have to be flat because then the cells adhere or they start sticking and growing to the medium, to this culture medium. Now you can clearly see when these cells from here are transferred to the culture vessel in the proper nutrient medium for its growth. After some days, when you look, or weeks together, you'll see proliferation, lot of cells growing. So the cells have, remember, keep in mind, in order for cells to grow, they must divide. So cell growth and division take place, and that's why you're getting so many cells in this culture vessel. Okay, so this culture vessel here, why is there growth? Growth cannot occur unless you're putting some growth factor. And that was the platelet-derived growth factor was added to this culture vessel. Clearly, you can see here no growth. Why? Because no PDGF. There was nothing to initiate the growth of the cells. Whereas here, we added platelet-derived growth factor. The moment, it's like, think about your enzyme degradation. Some of you worked with positive alpha amylase, you put it in the flask, and those who did negative control, a negative flask, were without the alpha amylase enzyme. You don't have an enzyme, how would starch be degraded or broken down, correct? Same concept. You don't put the PDGF, those cells will not be able to grow because there's no nutrient, no supplement for the growth. Here you added PDGF. Proliferation. We use this biological word or scientific word called proliferate. Cells proliferate, mean P-R-O-L-I-F-E-R-A-T. Proliferation means when the cells grow and divide. So you see a number of cells growing and dividing. Now, if you looked at these cells under the electron microscope, this is scanning electron microscope. So it's called the scanning electron micrograph. This is what fibroblast cells look like. Okay, under the SEM. I just put a larger image. Now, if you look here, these are intact cells, but you can see some of these cells here. Compare. Can you see these? Immediately, these are cancer cells. Because there's abnormal. Remember, cancer, as I explained in functions of mitosis, cancer is repeated abnormal growth and division. There's no stoppage. And we can see this abnormal growth taking place. So now, loss of cell cycle controls. What happens when there is loss of cell cycle controls in cancer cells? They do not respond normally to the body's control mechanisms. Cancer cells do not need their growth. Do they need? No. They just keep growing, dividing, growing, dividing. There is no stoppage to the growth and division of cancer cells. That's why I termed it as repeated abnormal cell division. So they don't require a PDGF to grow. Cancer cells do not need any of these growth factors to grow and divide. Why? Because they start making their own growth factors. They convey a growth factor signal. So what happens is many cells in our body try to mimic what we use scientifically, mimic, as though something is happening in the body. For example, some of the cells in our body may think, oh, this is what happens in a process called autophagy. Auto means cell, phagy means eating. Like cell eating, cell drinking. When we talk of endocytosis, I told you exocytosis. What happens over there? When you suffer from an autoimmune disease, all the white blood cells, which are the soldiers of the body, which build up our immune system, they are supposed to attack any foreign invader, which is a microorganism like bacteria or virus. But if our immune system is not strong, the bacteria or virus will kill our white blood cells, leukocytes, and infection. But sometimes our own white blood cells think, oh, this is a foreign microorganism entered into my body. Let's attack. And whom are they attacking? They are attacking and eating up other white blood cells in our body. That is autophagy, self-eating. What triggers our own white blood cells or leukocytes to eat others? I don't know. Why does it happen only in some individuals and not others? So they think, they mimic, they think something is coming and they start attacking. So that's what we're saying. They may convey a growth factor signal without even a growth factor being present. They may have an abnormal cell cycle control system. Definitely, it's abnormal for them to grow and divide. And that's what is happening in cancer. So we have to suppress that abnormal growth and division in cancer. And that's why you have chemotherapy, you have radiation, but now there is the new technique, which is the immunosuppressant technique, but it cannot be used for all cancers. So cells that acquire, we're going to be talking about DNA transformation in one of our labs to come. Understand the concept of transformation. Cells that acquire the ability to divide indefinitely are undergoing transformation. Cancer cells that are not eliminated by our immune systems, and then I explained all this to you. What's a benign tumor? I explained all these slides, students, do you recall? And I pointed out to this slide when I put it under my sub-heading of, I put it under the subtitle of importance of mitosis of functions, number 5. Okay, so I'm not going through it again. I encourage you to look through these cells. I explained these slides to you. So here, this last slide is just showing you, it's a culmination of all the stages of mitosis, including interphase and M phase, what exactly happens. Remember, in prophase, what happens? Meta, but pro-meta means beginning to come to the center, equatorial plane or center at the metaphase place. Ana means sister chromatids pulling apart, and telophase, you must have. What type of cell would this be, students? The moment you see this, what type of cell is this? Plant or animal? I called it invagination. Remember? Pulling inward.
Animal, because it's cinching.
Because it is?
Cinching?
Yes.
Cinching at the sides.
Okay, so what did we call that? A CF word? This is that CF word that is formed. Anyone? Anyone recalls? In animal cells, a blank plank is formed. In plant cells, a blank plank is formed. What is that C word?
Is it the cytokinesis?
Tanjima, yes, cytokinesis is occurring to
I remember.
form.
It's the cleavage furrow.
Perfect. Cleavage furrow. What in a plant cell? CP. And you eat in the
Cell plate. It's a cell plate.
cell plate, you eat in the plates. Okay, okay. I brought this. I showed it to you the last time, but if you want, if you would like, I can go through quickly. Would you like that? This is going to help you for tomorrow. Students, this is how exactly how you will see it under the microscope. So do we want to run through quickly? Yes? Okay.
Yes.
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 Come on. Let's see how much we remember our points. Okay, so I always start with this. Remember, dot, 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 dot. We called it the DG word. First word is D and G word. I said they appear as DG under the microscope. And that's why it helps you to identify the space. Look in your notes. What is the D one? 5-letter. 8-letter, second one. Anyone recalls? Okay, tell me which phase.
Blastular?
What's that? Ali?
Blastular?
No. Don't
said
look at
that.
that. Ali, these appear as
Dense?
yes,
Dense granules?
yes,
Okay.
under the microscope, and therefore which phase is it in?
It's not prophase. Or
I phase.
yeah.
Interphase.
Interphase, Professor?
Yes, interphase, because the cell has not begun cell division. Remember, G1, S, G2. Okay, let's look at this one. Even last time I pointed out to these. This, remember? I showed you all these, same phase.
Mm-hmm.
Look for my pointers. Okay, let's look at one. Okay,
Can I answer it?
yes, sure, Ali.
So for the top one, it looks like the nucleolus is still there.
Perfect,
So it's early prophase.
perfect.
Cool.
Nuclear envelope, nucleolus. And if you look here, nucleolus has almost disappeared. But first thing to tell you it's prophase, what are you seeing? These long structures called the C word. What is visible in prophase? First, chromosomes. Chromosomes visible, nucleolus is intact, and nuclear envelope. But if you look here, nuclear envelope is breaking down because you see spaces, not a complete outline. Nucleolus is small, and these are chromosomes. So I would go early and late.
Okay.
And here also late,
Mhm.
correct? Okay. This too is prophase. Because here your nuclear envelope is almost broken down completely. If I compare this one to this would be early and this would be late. See, I can't see an outline. No nuclear envelope or nuclear membrane. These are only long chromosomes, and a tad bit of the nucleolus almost disappeared. These are pointers, students, I came out with for you to enable to identify under the microscope. So look for these key criteria or points for tomorrow's lab, okay? Now, what would this be?
Which one? The squiggly one?
Yeah.
Prometaphase?
Good. Prometa, because it's not in the center completely. Center, no, it's a little away. So prometa, very good. Any metaphases here? Students, look all over and tell me if you see a perfect metaphase anywhere. Anyone is seeing a perfect metaphase?
Almost.
Where?
At
No,
the
perfect.
bottom.
Ali, I said perfect.
Perfect.
Because if you're saying almost, then it's prometa, correct?
Yeah.
I'm asking meta. I don't see. Maybe this one, but I cannot say for sure because it's at the edge. Okay, there is no meta. Okay, now let's go here. See, this is one cell. That is what phase? Yes.
Anaphase?
Very good. Perfect anaphase. Why? Because the sister chromatids have moved to opposite poles of the cell. They look at these little lines here. They are holding the sister chromatids at the kinetochore. What type of a slide is this? Plant or animal mitosis?
Plant.
Plant, because you can easily identify. Okay. Okay. What would this be? I pointed out these same ones. Look carefully. What with this? This is one. This is one. Same stage. Look carefully.
Is it telophase? Because
Yes, very good. Telophase. Why?
The
Ali.
center line,
What's that, Ali?
that line in the center
Correct. Called
of
what?
the cell plate.
Very good. The formation of the cell plate. So that's why I said, look very carefully, because sometimes, and I stated this exactly when I taught it to you, showed it to you last week, that sometimes we mistake anaphase, a telophase for an anaphase. But what's the distinguishing characteristic? You're looking for the formation of this cell plate, which you don't see in anaphase. Keep that in mind. I'm giving you a lot of clues to help you to identify. Okay. So with this, we'll finish. I'm going to stop this and we're going to go now to the next topic, which would be cell division only. So we're going to chapter 13. Okay. So I go back to PowerPoint lectures, chapter 13. And that would be meiosis, the second type of cell division, exclusively meiosis and sexual life cycles. Okay, so let's open that. You also open, it's always important for you to navigate it on Blackboard, so it's easier for you to understand. Okay, so let's stop my video so I can share it with you. I didn't share it. Okay. So we are seeing now meiosis and sexual life cycles. So we're looking at more or less the same, we're going to look, students, at the same organization of how of the questions, not all. First, we're going to talk about what is meiosis, when does meiosis occur, stages or phases, those three concepts of my whole summary questions that I organize my mitosis, and then we'll talk further. Okay. So what I'm going to do here for you is I'm going to now minimize this because now I want to open a new PowerPoint slide and I'm going to draw for you, okay, the what is meiosis. So just bear with me. And I'll explain to you what is meiosis with a diagram. Okay, students? Okay, so now, as soon as I put some more slides so I can, I'm going to explain to you with some more, just keep adding some more slides. Now I'll share it with you. And now we're going to begin from slideshow. Okay. So I'm going to explain to you now what is meiosis. Okay. So you draw this. not writing the topic, okay? So you know the top. Oh, I think it's too big. Let me erase this. Let me draw it slowly. And I'm deliberately going to draw in different colors, okay? So it is easily identifiable to you. Now take note, students. We use Roman numerals, meiosis 1, okay? And now I'm going to show you with a different color here. I'm going to show you with a different color, and this will be meiosis 2. Okay, I'm going to put 2n here. Now keep in mind this is meiosis, correct? So so this is 1n. This also is 1n, but I like to put, and I'll tell you why I put half an n. The books won't show you, but I'm trying to explain, okay? So don't get confused. So this will go through 1, and then this will go through. Okay, so students, what do I call this 2n? The same name for mitosis. What was this cell referred to as in my diagram? If you look back, what did we call this? Look at your mitosis diagram. What did we call this cell? Anyone?
Isn't that the parent?
Yeah, but we used the word. Give me the whole description, Draco. Very good. Original. What does 2n refer to?
Original parent cell.
No, 2n. 2n. What does 2n? Very important. What does 2n come from? What word? There's a difference. And I gave it to you in characteristics of living organisms when we talk reproduction. I introduced you to these words. The D word and the H word.
Oh, sorry. The 2n would be diploid.
So the original, and if you don't say this, it's absolutely incorrect. If you all don't specify diploid, haploid, you don't get points because that distinguishes, okay? So the original diploid parent cell, you can't just say parent cell, that's incorrect because you're not describing the parent cell to me. So original diploid parent cell, and then this becomes, what would these two be? These are 2 haploid daughter cells. And now here, these are called 4 haploid. Again, when you write haploid, you must write 1n daughter cells at the end of meiosis 2. So now I'm going to explain the diagram. You're going to write down the definition based on my diagram. So students, I keep stating this emphatically. If you use the book diagram, then you cannot go with my definition. My definition, my explanation match my diagrams. Are you understanding me? Because they won't show you half n, etc. So it is really pertinent for you to understand and comprehend what I explained to you. So let's go with, I'm going to use my laser pointer to explain now meiosis. Okay. So what is meiosis? Okay. Do you first want to write down the definition? Okay. So let's write down all the information I'm narrating to you or explaining in my definition stating is in my definition, is on the diagram. Okay, so just copy directly the words from here. So let's write down. Meiosis consists of two consecutive. Anyone needs the spelling for consecutive? Anyone requires the spelling? Okay, so meiosis, okay, thank you. Meiosis consists of two consecutive. Tell me what is happening here. The keyword is an R word. Can someone tell me what's the keyword? R word.
Reproduction.
No, another R
Reproduce.
word.
What?
No, not reproduction. That's my second question, Rico, when it occurs. Another R word. Look at the diagram. I'm sorry, I can't hear somebody's talking.
Reproduce.
No, no. No. Reduction.
Have COVID.
Please mute yourselves. Okay, so let's go here. So we are saying, see, everything is being reduced to 1/2, right? So what number would I write here, students? 2 times? Remember in mitosis, what number we wrote here? What does N stand for? What does N stand for here?
It's the nucleus, right?
No, N. N doesn't stand for the nucleus. This is the nucleus. What does this N number stand? Number of.
It's the number of chromosomes, right?
So 2 times what number you're going to write?
23.
Yeah, which is equal to, put all that information here. 2 times 23 equal to 46.
Okay.
I'm explaining every little detail to you. So it helps you when you're studying. Okay, what are you going to write here? 1 times? What is that? Yes.
Which is
23.
Very good. So it's equal to 23. Okay. Now here, I like to say half N and I'll tell you why. So this would be approximately 11.5, although we don't say 11.5, but I'm trying to show you everything is reduced to 1/2, because then 11.5 gives you 23. So egg is 23, sperm is 23. Okay. So let's go. So meiosis consists of 2 consecutive. The key word you're going to underline is reduction division. It consists of 2 consecutive reduction divisions, plural, meiosis 1 and meiosis 2. Or meiosis 2. Okay. Let's go now. Continue. The original diploid parent cell divides. Copy down from
So
the.
this goes after consecutive?
No. I'll repeat it, Tanjima.
It's Fatima.
Meiosis, what's that?
I said it's Fatima.
Oh, Fatima. Tanjima's the box's lighting. Okay, Fatima, I repeat the whole definition, okay?
Thank
The beginning. You're welcome. Meiosis consists of 2 consecutive reduction divisions, meiosis 1 and meiosis 2. And now we are continuing. This whole thing is the explanation, so I have to continue. Okay. Now we continue. Next sentence. The original diploid parent cell divides. The original diploid parent cell divides. Now, you can choose your words. I normally say to give rise, R-I-S-E, or you can say to produce. So to give rise to 2 haploid daughter cells, to produce or to give rise to 2 haploid daughter cells at the end of meiosis 1. Original diploid parent cell divides to produce or to give rise to 2 haploid daughter cells at the end of meiosis 1. Did everyone get those statements?
Yes, Professor.
Okay, thank you. Now we are continuing. See, I put all the information in my diagram, so it's easy. Okay, now we are going to say each haploid daughter cell of meiosis 1, each haploid daughter cell of meiosis 1 further divides. Each haploid daughter cell of meiosis 1 further divides. Again, keep the same word. If you say to give rise to, then write down to give rise to. If you wrote to produce, then write to produce 2 haploid daughter cells. Each haploid daughter cell of meiosis 1 further divides to produce or to give rise to 2 haploid daughter cells, resulting in a total of 4 haploid daughter cells each is producing 2 haploid daughter cells, resulting in a total of 4 haploid daughter cells at the end of meiosis 2. Did everyone get that, the whole definition, students?
Yes, Professor.
No, Professor.
Yes, what, Aline?
Can you repeat it, Professor?
Okay, which part? The whole?
Can you repeat the
Actually, from-
beginning,
Okay,
From the beginning,
okay, sure.
please?
So,
thank you.
sure, you're welcome. So let's follow the diagram because now I'm explaining too. Okay, so I said meiosis consists of 2 consecutive reduction divisions, reduction keyword, meiosis 1 and meiosis 2. The original diploid parent cell divides to give rise, RISE, or to produce 2 haploid daughter cells, at the end of meiosis 1 or meiosis 1. Each haploid daughter cell of meiosis 1 further divides to produce or to give rise to 2 haploid daughter cells. Each one is giving rise to 2, resulting in a total of 4 haploid daughter cells at the end of meiosis 2.
Got it.
Yes. So that's why, students, I give my own diagram with my own explanation, everything in the diagram. So it's easy when you are studying with the diagram. My definition matches my diagram. Now let's understand. So what am I saying? Think about, now we are asking ourselves our second question, because 1 and 2 questions go hand in hand, in my opinion, because I need to know. If I'd asked you a question right now, students, is meiosis taking place in your bodies? What is your answer?
No,
I
Okay.
because
guess.
oh,
Yes. Joshua, yes. Are you
Yeah.
saying?
In
What,
our reproductive organs.
no.
I don't know.
Are you
Isn't this-
pregnant right now? Are you pregnant right now?
I would say
I don't
no.
think so.
No.
I would say no because of the absence of sperm.
No. Okay, no. When is sexual reproduction? When meiosis is sexual reproduction, which is also referred to as fertilization that I gave you in characteristics. The union of the haploid sperm to meet the haploid egg cell to form the diploid zygote. So the answers are going to differ, right? Why? Because if someone is pregnant in the class, then they would say, yes, meiosis is occurring in my body because you're carrying the fetus. That's meiosis. You have the fertilized egg, the zygote, which is developing in the mother's womb, one function of mitosis. But if you're not pregnant, then meiosis is not taking place in your body. And there it goes. Mitosis was a 24/7 process, 365 days. Is meiosis a 24/7 process? No. You choose to reproduce sexually to give rise to the diploid zygote. That is meiosis. When sexual reproduction is taking place and the fetus or the zygote is growing, which would then be mitosis. So, however much we want to reproduce every day, you cannot. When you say, that's why I say do not use the word reproduction loosely. Because we tend to say, oh, my cells are reproducing. Are your cells reproducing? No, your cells are being produced. I always emphatically state to the students, there's a big difference between production and reproduction. Think of reproduction means you're giving rise to a whole new individual organism. Your cells in the body are being produced. And we need that all the time. That occurs during mitosis. But I choose to reproduce. Are you understanding the concept, students? And you do not reproduce every day. Who reproduces every day? A bacteria through binary fission because they are reproducing asexually, not sexually. So it is important to understand now what happens to this egg. Okay. Why did I say one end? Because when does meiosis occur? My second question, during sexual reproduction or fertilization. And we know what it is. Haploid sperm swims to meet the egg, unites with the egg to form the diploid zygote. Now, what happens to that zygote? That zygote is 2N, remember? Why? Because egg brought her 23 chromosomes, sperm brought his 23 chromosomes. Therefore, the zygote, which is diploid, 2N, 46 chromosomes, has a combination of maternal, mother's, and father's chromosomes. That is why we are so different. Because what are on chromosomes? I said genes. And genes specify our traits, genes determine our traits. I gave you all this under structure of a chromosome. And that comes in meiosis. Okay, so I'm going to now stop this. Therefore, this egg will go through what we call oogenesis. We can write that word down. Let me stop my laser. Okay, and I'll go to the next slide. And so we call this oogenesis, OOGENESIS. What is oogenesis? Oogenesis is the process of the production of haploid egg cells. Please write 1N. Okay, I'll write it here. Of haploid egg cells. Where are our eggs made?
The ovaries.
Yes, so I'm writing, this is female sex organ. In the female sex organ called the ovaries. I gave you a full descriptive definition of oogenesis. OOGENESIS. And the next word is called spermatogenesis. Who can form the definition for me? Who wants to try? What's the definition? Keep in mind, the same definition I gave you? Who wants to try?
I'll
I'll
try.
try.
Oh no, go for it.
Okay, go ahead, Alan.
No, it's okay. It's okay. You go.
Who's
Probably-
that?
When the process of pre-production of the
No reproduction.
reproduction,
No reproduction. Process of production.
the production of haploids in the sperm of a male sex organ is called. Told you I'll probably get it wrong.
No, good try. I'll repeat, okay? So, oops, where is it? Yeah. Trying to use my laser. Okay, so we're going to write spermatogenesis is the process of the production of the haploid sperm cells in the male sex organ. You all know how to write male symbol, students? Important. In the male sex organ, it's a circle with an arrow called the testes. ES is plural, IS is singular. So again, I repeat, spermatogenesis is the process of the production of the haploid sperm cells in the male sex organ called the testes. Okay. Now, let's go here, and we're going to now look at meiosis. So we finished with what and when, and many of the slides I've arranged under mitosis, structure of a chromosome. Okay, so we've talked about what is meiosis, what is heredity. It's the transmission of traits from parent to offspring. I used it in genetics under structure of a chromosome. All these I put under my title in mitosis. So students, I want you to take note, make a point of reference. This slide and many more slides come under structure of a chromosome. So you're going to say, refer to structure of a chromosome in mitosis for all definitions. I'm being very explicit, okay? So, and I explained what genes determine or specify a trait. So we are seeing here, this is the father, this is the mother and the kids. And you can see what the children are. The children have a combination of both maternal and paternal traits, right? And that's what we're looking at in genetics. We know what are genes. All these I've put in, again, I repeat, under structure of a chromosome. But one thing, when you talk of gametes, I keep saying gametes are sex cells. How many gametes do we have, students? What is your answer? How many gametes do you have?
Well, men, 1 million.
Four? Did someone say 4? I don't have 4. I have 1 gamete, 1 sex cell. What is my sex cell? Egg cell. What is the male sex cell? We just did it. Sperm cell. We can't have 4. That's abnormal. You have 1 gamete. Gamete is a sex cell. I cannot say I have an ovary. That's not my gamete. A gamete is a sex cell. My egg cells are my sex cells. The sperm cells in males are their sex cells. There is a difference between cells and organs. Male sex organ, testes. Female sex organs, ovaries. Where are our ova? OVA is a scientific word for eggs. Where are our ova made? In the ovaries. Where is sperm made? In the testes. So you have to be able to distinguish the terms, okay? Now, all the cells in our body are referred to as somatic cells, except our sex cells. And all the cells which are the somatic cells, which are the cells in the body, have 46 chromosomes, except our sex cells, which are haploid, okay? What is a clone? A group of genetically identical individuals. I've talked about loci or locus under structure of a chromosome. We've talked of sexual reproduction, fertilization. Remember, I pointed out to you in mitosis, I came here and I opened how Hydra reproduce asexually through bud formation. And I pointed this red ones. Now, all this again, I repeat, all this I have structured and compiled my information under structure of a chromosome. These are all very important terms. Now, take note of this word called homologous chromosomes. When I bold it, when I highlight it, they are important terms. Homo comes from the word same, like Homo sapiens. Homologous are chromosomes that have the same size, shape, and banding patterns. And I've put it here. All this again comes karyotype under structure of a chromosome. So I've explained. I need to point out, and I think I pointed out to you when we did mitosis, I came and opened the slide. This illustration. Now, look carefully at the karyotype and explain to me what type of karyotype is this. I think I brought it out to your attention.
You asked what type of karyotype?
Yes. Look carefully at the karyotype. Look carefully.
Identical?
No, no, no. No, look carefully. Remember I said, what does a karyotype contain? What does a karyotype have? 23 pairs of chromosome. What are those 22 pairs called? If you look at structure of a chromosome, irrespective whether you are male or female, the 22 pairs I said were called the A word. If you look
Autosomes. Autosomes.
what differentiates me from a male? What differentiates me from a male?
The sex chromosomes?
Very good. What is the sex chromosome here?
Should be X and Y.
Very good. So
X.
what would you call this?
That's a male karyotype.
Yes. So how are you going to answer in the exam? You're going to say, this is a human. I brought that out last time too. Very important. This is a human male karyotype. If you just say karyotype, incorrect. That means you were not able to identify it. So look at everything. Look at the 23rd pair. You go straight to the 23rd pair. Why is it male? Because of the presence of the
X and Y sex chromosomes.
Y chromosome.
A Y chromosome.
What did I give you about the Y chromosome in the notes? All this is given. I have given you such detailed notes. Read, students. Every note, every sentence in the note is important. What is so important about the Y? Look at the Y in the diagram. And Joshua, you had pointed out about the Y chromosome, correct? But what do we see? What is so pertinent about the Y chromosome? What is so important about the Y chromosome, students? Look in the notes if you don't know. No one can identify what happened. The Y chromosome is shorter than the X, and therefore it carries lesser number of genes. Extremely important. That Y determines the male sex, but it's shorter and carries fewer number of genes. So when you look here now for the maternal and paternal, we see very clearly that the diploid zygote gets one set of chromosomes from the mother and one from the father. But here, what are we seeing? We're seeing that the sister chromatid, the moment you see this, that's the centromere here, the point of attachment of the sister chromatids, as in anaphase. And these are pairs of homologous chromosomes. Very important. Homologous chromosomes are only seen during meiosis, not during mitosis. Okay? So we see here all this information, 22 pairs, autosomes, 1 pair, sex chromosome. All this I've already pointed. I keep repeating myself. This is all under the subtopic structure of a chromosome that I've given you. Now, from here begins the variety of sexual life cycles. So there are a number of sexual life cycles that occur. The alternation of meiosis and fertilization is common to all organisms that are going to reproduce sexually. So there are 3 main types of sexual life cycles, but these life cycles are going to differ in the timing of the meiosis and the way fertilization takes place. You will be getting questions. You could get questions. Mark the slide important. So we are making a comparison between animals, plants and some algae, and most fungi and some protists. So when you look at animals, what do we go through? So how is the zygote formed? The haploid egg and sperm cell, the 2 haploid gametes undergo fertilization to give you the diploid zygote. That diploid zygote grows and divides and undergoes mitosis to give you the diploid organism. And then it again goes through meiosis, which is haploid gametes to give you diploid zygote. Okay, so we have a haploid and a diploid stage. A diploid is mitosis, haploid is fertilization or meiosis. Let's look at what happens in plants and some algae. Now, here, the haploid multicellular organism is called the gametophyte. So they have 2 life cycles. They have a gametophyte generation and a sporophyte. Remember, where did I use this word spore formation in Characteristics of Living Organisms, chapter 1? Can anyone recall with what reference I talked of spores? I said not all plants reproduce sexually. Some reproduce asexually. Remember? Which was that particular plant? F word. Do you all recall anyone?
A fern?
Fungus.
Good.
Fungus.
Fern. F-E-R-N, fern plant. Remember I told you if you look at the undersurface, you get round brown little spots. Those are spore formation. So they reproduce through spore formation. So you see, gametophyte would be the gametes. And the gametophyte generation is haploid, but the sporophyte generation is diploid. That's what you need to know in plants and some algae. Let's look at fungi and some protists. In fungi and some protists, haploid unicellular or multicellular organisms. So the mitotic stages would be what? Haploid. And what would be the gametes also are haploid, right? To give you the diploid zygote through fertilization. So haploid unicellular to give you diploid multicellular organisms in the zygote. So both meiosis and fertilization is going to be haploid, and the zygote will be diploid. Okay, and all that explanation is here. So this is the definition which I gave you first. I explained what is meiosis, correct? Reduction divisions. So we finished with that slide. Now, what I'm going to do is 12:30 is my point of giving you a break, right? So It's 12:30. We'll come back in 20 minutes, students. I'm going to stop sharing this slide. So we'll have a break. I'll see you back at 12:50.
Thank
So,
you.
12:50.
Thank
Enjoy
you.
your break.
Thank you.
Oh, you're welcome. Hello, students. We are back. Hi. Okay, so I'm going to now share again, right, where we came. So now this is important, okay, students? What I want to say is I want you to focus on all stages or phases of mitosis, but in meiosis, and particularly prophase 1 of meiosis, most of the events are occurring. So it's a very important phase for you to remember. So I'm going to lay emphasis on this prophase 1 of meiosis 1. So now remember, just to reiterate, we know meiosis consists of two consecutive. Consecutive means one after the other. Reduction divisions, meiosis 1 and then meiosis 2. So in the diagram, the original diploid parent cell went through, divided to give rise to two haploid daughter cells at the end of meiosis 1, as my diagram shows you. But what are we saying? It is going to divide to give rise to two haploid daughter cells. That means those two haploid daughter cells were formed only after the parent cell, diploid parent cell, went through prophase 1, metaphase 1, anaphase 1, telophase 1. So cytokinesis had to occur, which is the division of the cytoplasm, and karyokinesis to give rise to or to produce the two haploid daughter cells at the end of meiosis 1. Now, those two haploid daughter cells of meiosis 1 further divides, and it will go through prophase 2, metaphase 2, anaphase 2, telophase 2, resulting in 4 haploid daughter cells at the end of meiosis 2. I'll put it into perspective. So now, think of the PP mat, right? Prophase, prometa, meta, ana, telo. Same things that happen in mitosis are occurring in meiosis 1. But keep in mind, the difference was we never saw homologous chromosomes in mitosis. Homologous chromosomes are only visible or seen during meiosis, sexual reproduction. So these are key points and terms you must remember. Okay, so now let us see what are the important events that occur in prophase 1 of meiosis 1. So in early prophase 1, each chromosome is going to pair with its homologue. The first thing that happens, students, is crossing over. Now watch my hands. My hands, say, represent a pair of homologous chromosomes. This pair of homologous chromosomes cross over. What is happening when my hands are crossing over? What happens here? What is happening to my hands?
It's touching.
Perfect. It's touching. And what do you mean by touching? Therefore, I'm trying to explain. What is happening when it's touching? There is
Bonding. Bonding together.
Okay. No. So you see, yeah, but I wouldn't call bonding. So when it's touching, this part of this hand, of my right hand, is meeting with this part of this left hand. So there is exchange of genetic information taking place. This is the X, you know, where they are meeting is called an X region called chiasma. And so if this My hands represented a pair of homologous chromosomes. There is exchange of genetic information at this X region, which is this intersecting point called chiasma. And I'm going to point that out to you. So X-shaped region called chiasmata or chiasma are sites of crossing over. And when they are crossing over, remember, there is exchange of genetic information taking place. So now, okay, you know what I'm going to do? I'm going to do a new share and I'm going to go here and show you. Okay, so I'm going to deliberately draw my homologous chromosomes as in different colors so you can see it carefully. So this is a pair of homologous chromosomes. Where now the homologous chromosomes are intersecting at this point, correct? They're meeting here. So this meeting point is called chiasmata or chiasma, which is that X-shaped region. This X-shaped region. And at chiasmata, I said, there is genetic information being exchanged. So now, what is the homologous chromosomes containing? Maternal from the mother, paternal. So at chiasmata or chiasma, the X-shaped region, maternal, paternal chromosome genetic information is exchanged. That is, the meeting point is also referred to as what we call synapses. And so we're going to see now, all this occurs only during meiosis 1 of, during prophase 1 of meiosis. So let's see, put it into perspective. You had the pair of homologous chromosomes. We see this is maternal, paternal. They duplicate, so sister chromatids, because you see the centromere formation, the diploid cell with the duplicated chromosome. See, this forms duplicates, this duplicates, so you have a pair. And now in meiosis 1, the two daughter cells are haploid because each one is receiving one copy. They separate out. So two haploid daughter cells, one paternal, one maternal of the duplicated chromosomes. And then you have four haploid daughter cells. So one separates, one separates, one separates, one separates. And so you have two, remember I said going to form the sperm cell, blue, paternal, maternal X cell. So each will have one set of chromosomes from the mother and father. That is the diploid zygote. That's why everything has to be reduced to 1/2. If X cell brought her 46, sperm cell brought his 46 chromosome, what would we be? The diploid, we were once zygotes. We would have 92 extra. I just describe them as extraterrestrials, ETs, right? What are we? 92 chromosomes? I don't know what we would look like, right? Because we only need 46. So everything has to be reduced to 1/2 in meiosis. And that's what we are stating emphatically. Now think, what happened in meta? Meta came from the word middle or equator. So again here, the pairs of homologs, because you're not having one, yeah, they are always homologous chromosomes. So pairs of the homologs, homolog is one of the pair. So pairs of the homolog, they move to the center of the cell at the metaphase plate. Microtubules from one pole are attached to the kinetochore. You knew the spindle fibers holding it at the kinetochore, at the centriole pair, only in animal cells, not in plant cells. Anna, we knew what happened, sister chromatids, but pairs of the homologs are separating, moving to opposite poles. Okay, sister chromatids remain attached at the centromere, move up as one whole unit towards the pole. Telophase and cytokinesis, what happened? Now that one cell has to divide. So cytokinesis, division of the cytoplasm, division of the nuclei, and each chromosome still is consisting of the two sister chromatids. And they now have to split, right? So that each daughter cell receives one set, right? So no chromosome, one keyword, no chromosome replication occurs between meiosis 1 and meiosis 2. There's no chromosome replication because the chromosomes are already being replicated. Okay, so when you look here, look, maternal, paternal. These are the homologous chromosomes. Exchange of genetic material takes place at the X-shaped region called the chiasmata. That's in prophase 1 of meiosis 1. Metaphase, what happens? This homologous pair is going to move to the center, as we just said, at the metaphase place. There is an arbitrary line if you look here. What happens here? Now, look carefully. In anaphase here, the sister chromatids. Now look, this is paternal. This is maternal, mothers. But it has, because of that exchange that's taking place over here, now part of the chromosome is from the mother, exchange with the father. Part of the father is exchanged here. And so immediately that tells you that occurred in prophase 1, meiosis 1, synapses. Then look here. This is, again, mothers, maternal and paternal part. Paternal and paternal. So there's exchange of genetic information. That is why, students, we are all not alike. Our DNA is different. Our genetic composition is different because of the exchange that has taken place during chiasmata at synapses in meiosis 1. And then you see cytokinesis, cleavage furrow formation. You see, this is the mother's maternal, father is blue. And partly because that's where synapses occurred. This is where synapses occurred. You have the paternal with the maternal. So immediately when you see a homologue or a homologous pair with different colors, you know immediately that should be a clear indicator to you that synapses has occurred, which is exchange of genetic information in that X-shaped region where synapses, chiasmata has occurred during synapses. Okay. I'm not, you can just browse through prophase 2, metaphase 2. Remember what has to happen in prophase 2. Now remember, you had the 2 haploid daughter cells at the end of meiosis 1. So what is happening now? The spindle apparatus forms. Late prophase 2, chromosomes, it's still composed of 2 chromatids, move up to metaphase plate. Metaphase, what is happening? Sister chromatids are arranged at the metaphase plate. And because of the crossing over that occurred in the meiosis 1, the 2 sister chromatids now of each chromosome, they are no longer genetically identical because they exchange the genetic information. Okay. And anaphase 1, sister chromatids now move as 2 newly individual chromosomes to opposite poles. And telophase, the nuclear form, karyokinesis, and chromosomes begin to decondense, shorten and thicken. Okay, so that's what they're showing you here. See, students, the key concept, if you know mitosis, what happened in each stage of mitosis, keeping in mind and putting into perspective, meiosis 1, everything has to be reduced to 1/2. Meiosis 2, everything has to be reduced to 1/2. But what should happen at the end of telophase 1, telophase 2? You must end up with daughter cells because cytokinesis, karyokinesis. You see, textbooks don't show you karyokinesis, but it's important for you to understand. Okay. See, I kept talking, crossing over 1 and synapses occur during prophase 1. During synapses, what happens? The DNA is going to break. DNA breaks are repaired. Now, later in our chapters towards the end, 17, 18, 19, we'll be talking about, we have repair enzyme. When I explain DNA replication process, students, a lot of things can go wrong when our DNA replicates itself. And at that time, I'll be speaking about a particular enzyme, repair enzymes. We have enzymes that can repair the excision. See the biological term I'm using, EXCISION. To excise is to cut off. a break that has occurred in the DNA. So that's where we are talking about in synapse. That's what is occurring in synapses. During synapses, which is that meeting place at the chiasma, DNA breaks are going to be repaired. So if part of my DNA is broken, that repair is occurring during synapses. The joining of that segment of the DNA which has broken off, joining the DNA from one non-sister chromatid to the corresponding segment of another. So this is key. See, I highlighted the word. So we are re-emphasizing the events that occurred in meiosis 1. Synapses and crossing over in prophase 1, very important. See, that's why it's highlighted here too. Homologous chromosomes physically are going to connect, right? And exchange the genetic material. Homologous pairs of chromosomes are at the metaphase plate and separation of homologs during anaphase. All these are the three main steps that occur in meiosis 1. So I've just shown you a distinction between, or a distinguishing features or comparative study that you're making between mitosis and meiosis. So I encourage you to go through that diagram. And I've just given 2 sheets exactly what happens in mitosis and meiosis, what happens during replication, number of divisions, etc. So just go through those slides. Now, over here in this slide, I love mitosis, meiosis. I get very excited to teach you these topics. I hope you are enjoying these topics. So when we talk of genetic variation, we've talked about a mutation in chapter 1, if you can recall. What's a mutation? A mutation is a sudden change brought about in the gene structure. Now, always keep in mind, mutations, when we think of a mutation, at least when I'm thinking, I'm always looking at a harmful mutation. No, there are both harmful mutations and there are beneficial mutations. So we need mutations to take place. Otherwise, we would all be identical. So those are the beneficial mutations. So they are the original mutations, are the original source of genetic diversity. Now, I want you to write down 2-3 sentences, which I'm going to culminate from this slide. Okay, write this down. In my opinion, they are important. So write down. Meiosis results in, and I probably have given it to you, genetic variation. Meiosis results in genetic variation or reshuffling of genes or alleles, reshuffling of genes, or so genetic variation, reshuffling of genes, or what I said, reshuffling of genes or independent assortment. So it results in these 3 phrases. Now, think of what I'm saying, students. I just said assortment. There is independent assortment of genes taking place.
Assortment
of genes. So we say genes assort themselves independently of each other. We're going to put this is a law, which we're learning in the next chapter. It will come again, law of independent assortment, which Gregor Mendel, our father of genetics, put forth. That's our next chapter, Mendelian genetics. I like to call it Mendelian genetics. So let's understand. Think about it, students. If we did not go through meiosis, we would not produce offspring or progeny, correct? We would say goodbye to our family name. Secondly, if we did not undergo sexual reproduction, there would not be any genetic variation taking place. All individuals would look alike. Why? Because the haploid sperm brings his 23 chromosomes, haploid egg brings her 23 chromosomes. So we were all once diploid zygotes, which had a combination of maternal, paternal, egg and sperm chromosomes, which is nothing else but meiosis. So because on account of meiosis, there is genetic diversity. We are so diverse in the genes that we possess. And therefore, genes specify or determine our traits. And that's why we are so different.
Can I ask you a question?
Sure.
So for organisms that don't reproduce, or like that don't need a partner to reproduce, the genetic diversity in their species then is really only attributed to like just mutations in that one, organism's DNA that,
Is Ali asking me the question?
Yeah.
Okay, Ali, so when you're saying organisms that don't sexually reproduce, they will reproduce asexually, and the only organisms that can reproduce asexually are bacteria, viruses, and some fungi and plants that undergo through spore formation. Correct?
Yeah.
Now plants reproduce, most plants reproduce sexually.
Mm-hmm. Oh, okay.
Because they have the male sperm cell and the female egg cell, which does not look like ours,
Mm-hmm.
but we also refer to it as sperm and egg cell,
Mm-hmm.
which goes through fertilization.
Yeah.
Now, all higher organisms must reproduce sexually. So you need an egg cell and a sperm cell to form a diploid zygote.
Right.
You can't have two egg cells or two sperm cells forming a child, right?
Mm-hmm.
In humans or in complex organisms, no. So what I'm saying is, does it mean that there won't be any genetic diversity? Yes.
Mhm.
Remember, meiosis results in genetic diversity because we perpetuate our own kind of species. I'm giving rise to only a human organism, right?
Mhm.
So if I don't sexually reproduce, then I am not producing children,
Mhm.
progeny. So my children won't be able to produce any more children, right? And this is a choice. So we are ending our own family tree,
Mm.
right? But
Mm.
I was a zygote. I got one X chromosome from my mother, from her egg cell, and one from the sperm cell. That's why I'm a female. Now the males get one X from their mothers, but their Y, only the males have Y. So the fathers, the sperm cell had to give them their Y, and that's why they got XY. And we see what are on these chromosomes? Hundreds of thousands of genes, which I put under the subtopic, which I keep emphatically stating, structure for chromosomes.
Yeah.
And a gene is a unit of hereditary information that is being transmitted from parent to offspring,
Mhm.
from my parents to offspring. Now, when I choose to undergo meiosis, sexual reproduction, I am then passing on my genes.
Mhm.
You have not inherited traits from your parents, you have inherited genes.
Right.
And those genes that you inherit from the parents determine your traits, like hair color, eye color. And it's fun because in genetics in the lab, we will be looking at the traits we possess. And you may not even know that you have them till we learn them. And you'll say, oh, really? I did have this trait? I didn't know. So that's a fascination of genetics. And we're going to go into the segue of next topic, wherein we'll see it was Gregor Mendel, the father of genetics, who put forth, what are these genes? How do we inherit? What type of genes are we inheriting from our parents? Why don't they show up? But geneticists do not know the exact number of genes on each chromosome. I want you to think, we have 46 chromosomes, 22 pairs, autosomes. All of us have the same 22 pairs. But that one pair, the sex chromosome, distinguishes the male from the female.
Right.
And each chromosome, if we have hundreds of thousands of genes on one chromosome, think we have 46 chromosomes. How many genes are we having? So what happens in meiosis is, we say there is reshuffling of genes taking place. Meaning, I use the analogy, when you play cards, you have a deck of cards, you shuffle the pack, you distribute, say, a deck to each individual, right? So you pick up your cards. Okay, you play another game. You may have lost or won the game. You play another game. You then shuffle the deck of cards, the pack of cards. You give out a deck, 10 cards. Does that individual have the same deck? No, it's all random because there's shuffling of genes. So what geneticists say is, what type of genes are we inheriting? The genes that we inherit are on account of external factors that control them. Now, we geneticists, when we go into genetic disorders, I love these topics. I'm very passionate about genetics and genetic disorders. As I don't know if I think when I introduced your first day of classes, I said I was privileged to be chosen for one of the 30 people in the US to attend the Human Genome Project, wherein Dr. Francis Collins from NIH, National Institute of Health at Bethesda in Maryland, runs the Genome Project. And they have mapped, yes?
Yeah, I've heard of it. That's
yes.
really cool.
And they have mapped all our 46 chromosomes. And I was privileged to see mapping. And when you look at the length, I was flabbergasted. Length of one chromosome is like, I always look at, you know where our lab is, right? The science department. The whole of the length of the north side of our 6th floor is the length of one chromosome. So imagine you have to envisage, oh my God, if one chromosome is so long, hundreds of thousands of genes on one, and I have 46 chromosomes. Look at the plethora of genetic information I carry. But then what am I passing on? I'm passing on some of those hundreds of thousands of genes. And so all these processes are occurring, crossing over, random fertilization is taking place. That's why when you look at your siblings, no two siblings are identical in their DNA, even if you are identical twins. Why? Because of random shuffling of genes. And I'm looking at this. Next slide. What I'm speaking about. In independent assortment, each pair of chromosomes, one from the mother, one from the father, are going to separate, and the daughter cells have a combination, right? Maternal, paternal, red and blue, I showed you. Now for humans, think about it. N is 23 because we have 23 pairs of chromosomes. How many? There are more than 8 million possible combinations of chromosomes, of genes on the chromosomes that we are inheriting. So every time your parents produced a child, your child, the offspring, which is your sibling, picked up. Now, many of you may be having children in the class. You see your spouse or significant other gave his 23 chromosomes, you gave your 23, and your child now has a combination. So some of the traits look like your significant other, your spouse, and some look like you because of the random shuffling of genes. Environment plays a vital role as to which genes the child has inherited from either parents. And also, we see it's on account of the crossing over during meiosis, sexual reproduction, that is producing recombinant chromosomes. Genes, remember, maternal had paternal, paternal had maternal genetic information because of the crossing over at synapses. All this results in genetic variation. That's why we are so different in our traits.
That's crazy.
So isn't this fascinating? So crossing over contributes to genetic variation. And in humans, an average of 1 to 3 crossing overs occurs per chromosome. So anywhere between 1 to 3, and we have 46 chromosomes. Every time the chromosome crosses over, it can cross over 1 or 3 times on an average, 1 to 3 times. And we
Can I
have
have a
46.
question? Sorry to interrupt you. It's just very interesting. So I had a thought. Do you know if there's any correlation of like how many times those genes cross over with like, I don't know, I guess whatever qualities that person has, you know, if that impacts like, I don't know, like what they're good at or how many things they can do, or I don't know.
I don't know if I'm understanding you, but I'll try my best to. So for example, like you want to, I don't know if I'm putting I understood your question, but I'll just try to put it into perspective. So that's Ali, right?
Yeah.
So Ali, are you looking at, so for example, if you have an artistic trait, you're good in art and drawing, and
Yeah.
your sibling may be good in playing an instrument.
Yeah.
Okay, so immediately, what do we ask? We say, oh, is your mother or father an artist? Don't
Yeah.
we say that?
Yeah.
Yes. Yeah. So immediately we can now identify whether you have inherited. So let me take my example. Okay, my father was a doctor and my mother was a teacher. God bless both of them. They both passed away. My father always wanted me to carry on his medical practice. My mother wanted me to be a teacher, educator. Now, yes, I'm not a medical doctor, but I am an educator, but I'm also a scientist, and I'm so passionate about the medical field. So I inherited genes from both mother and father, but fortunately, my parents never pressurized me. Like my father never said, you have to be a doctor. He gave us a choice, whatever we were passionate in to choose. So I'm looking to see now, yes, I have picked up genes from maternal and paternal side, strong genes, because in my family now, most of us, and my mom's side, most of her sisters are all educators. In my father's family, most of his family members are doctors. So we have a culmination of both medical and educators and running in the family. My sister's an educator, my niece is an educator, my, nephew is a biomedical engineer. So, brother-in-law is a chemical engineer, sister was a chem. So we have those genes running. So you see, you can tap into genes. Now, I was just having a conversation earlier with one of my older sister, and I said, you know what, we've never really, I don't know whether I've exploited most of my talents. We don't exploit to the maximum our talents, right? I always say to you when I draw on the board or I'm drawing on my screen, I am not good in drawing, but I force myself to draw. But I've never tapped my talent of artistry. I've never tapped other talents. So we have those hidden talents, but there may be a point where you see, you're very good in art or you're very good with your hands. And you suddenly, decorate things and you say, oh, wow, I never knew I was so good in table decorations or origami, right?
Yeah.
So, and sometimes it's not only your parents, but this is the fascination. And that's what we're going to learn in genetics. Genes skip generation. So your great somebody must have had that talent and now it's shown in your generation.
Crazy.
I hope I understand.
No, that was my question because I was just curious about like.
Yeah, and sometimes
the qualities like talent.
Yes. Yeah. So, if you look carefully and, normally when I'm teaching, it's always during Thanksgiving break or spring break. And I say, okay, students, especially Thanksgiving break, when you're sitting at the family, at the dining table and having, Thanksgiving dinner, look and see. Oh, especially because we do the genetics traits, look and see if you have detached earlobes. Who has it, your mother or your father? Or if you have we'll be doing widow's peak Or if you have, like, you look at my hands, okay? I'll show it to you like this. Okay, here. Can you see my fifth finger? It's completely bent. Can you all see? So this is bent little finger, which is a genetic trait, and it's a dominant trait. Now, most of you will have straight fingers, but mine is completely curved, both hands.
Mine is too.
So that's bent little finger. Now I cannot see which of my parents had it. They're not living to see it. And I wish I knew whom I inherited it, from my father or my mother. But I'm looking at their photographs here in front of me teaching. I've got all their frames. And I see my mother has a typical, very prominent widow's peak. So I've definitely inherited it from my mother. The widow's peak. I'm looking at a photograph. Clear widow's peak she has. So you see, when you look at the traits, immediately someone will say to you, oh, you look just like your mother. They say, I look like my mother. I know I sound like my mother because many colleagues have called up and they've spoken to my mother when I was in the college and during the conversation. And I always say this to my students. During the conversation, my mother had to say, oh, I'm not Manita. I'm not Manita speaking. I'm her mother. And then they would stop the conversation saying, oh, I'm so sorry. But you see, this is because of the dominant genes you all have inherited. It's fascinating to see what you have inherited. What if you have kids, students, you will see what you have passed on to your own offspring. Traits you have passed on. So this was the chiasma that occurred, X-shaped region. Let me just finish this and we finish this topic. So they're just showing you prophase 1, metaphase 1, and this. So with this, we finish this. I hope this was interesting. And we are going to start with, I'm just going to show you here. Oh no, stop sharing. We will start with the next chapter, which is, let me just go to my PowerPoints. Our next chapter will be Mendel and gene idea, which is Mendelian genetics. So that's our segue. It's fascinating. We're going to go into more and more genetics out here. I hope you enjoyed my class. So
Yeah,
tomorrow we will go into mitosis, stages of mitosis, okay? Mitosis, meiosis. So it would be, I don't know, I get very excited. You watch in the lab, I'll be showing my enthusiasm and excitement when I see the stages of mitosis, especially when you make your own onion root dip, okay? I'm going to call it a day and not go into start a new topic today and leave half of it. So we'll start with, I'll just write this down. I like to write where we are going to start with the next class. So next class, we're going to begin at chapter 14, okay? So anyone who came in after I had taken attendance, students? Let me see, okay, I'll call out. Is Alan here? Alan? No, absent. Is Errol here? Errol?
Yes.
Okay. Is who else? Brittany not here. Who else? I didn't mark. Okay, everyone else I have got. Michael, of course, Michael is not here. Tanjima, yeah, I marked. Michael is not here as yet. Okay, so I guess he's on jury duty. Okay.
Professor, did you mark me?
Who's that?
Andrea.
Andrea Saltos, your last name.
Yes.
Yeah, I think I marked you absent. Okay. Andrea. Thank you, Andrea. Anyone else? Pamela, no, I marked her absent. I haven't seen Pamela. Okay. Have a good one. Students, take care. I'll see you all tomorrow. Aline, you can wait. Who wanted to ask me a question? Was it Aline, Reiko? Somebody wanted to talk to me? Reiko, after class,
I have
right?
a question, Professor.
Sure, go ahead.
I was about the lab.
Okay, then come in, officer.
Okay, thank you.
Is it just 1 minute question or you need explanation?
Maybe like 5-10 minutes.
I enjoy, is that who's this speaking?
Brandon.
Brandon, come in,
Yeah.
officer Brandon, so I can explain to you.
Okay, thank you.
Like last week you came in, come in. So anyone, has questions, you'll like a group, y'all came in and it is, y'all can all benefit if you have any questions. Brandon, I'll see you in officer, 2 o'clock.
Yeah, thank you.
Okay, you're welcome. Have a good day, students.
Have a
See
nice
you.
day.
Professor.
You too. Thank you. Very
You too.
interesting. I
Bye.
Bye-bye.
Bye.
Bye.
Dr. Pavel,
Yes, Ali.
I want to say it was very interesting. Genetics is pretty cool.
Yeah, I
Explain.
hope. Ali, what's your majors?
Actually, so I'm taking this biology course just because I enjoy science.
Oh,
I'm gonna be a lawyer.
oh,
Yeah.
so
So I'm
patent
in
law.
the Hunter for poli-sci, and then I'm gonna go to law school. But yeah, I just really like science and biology.
Ali, sorry to interject. You
No, it's
can
okay.
you can do patent law with this science background,
Oh, cool. That'd be cool.
right?
Yeah.
Yeah, because you seem very intrigued in biology, and you're keeping abreast with it. Very good.
Thanks.
Yeah,
Really cool. I didn't even know that.
yeah. Good
Cool.
luck. Good luck.
Thank you. I will need it.
Don't worry.
See you later. Thank you.
Keep your questions coming. I'm more than happy to take questions.
Yes, ma'am. I
Take
can
care.
promise.
I'll see you tomorrow in class.
Tomorrow. Thank
Have
you.
a good one.
You too. Bye.
Bye. Angel wants something? Angel? Angel?
No.
No. Have a good one, Angel. If you have any questions, please don't hesitate to come during office hour with the lab report questions or anything. Okay, Angel?
Okay. Thank you.
Have a good one, Angel. Take
Yeah,
care.
you too.
Bye-bye.
Thank you.
Hi, Reiko.
Hi, Professor.
Yeah. What's going on? Will you? So
I
you're
have
not-
a cat that I had my cat for 19 years
Oh,
and she's dying.
oh.
And I
Oh,
don't want to leave her in the
Oh.
house while I'm going to school tomorrow.
tomorrow.
And then
Okay.
coming back and watching her die.
No.
You know what I mean? It's just a little emotional for me.
Yeah, yeah.
But I did all 3 of those labs that we needed to
Okay.
do,
Do
all
me-
the graphs.
Okay, do me a favor. When you come in the following week, be very discreet and just hand it to me on my folder. Just keep, you know how you come up all the time, no?
Yeah,
Bring it to me and just give me.
okay,
Don't worry. Okay?
all right. I kind of really wanted to do that onion
Yes.
thing.
yeah.
I really
Good
want
tip.
to see it, but I don't want to leave her, Professor. I really don't
No,
want to
no,
leave
no,
her. And
no.
then I come home and she's dead, and
No,
she
no,
died
no.
by us. I
What?
can't.
What happened? But 19 years? Wow. God
Yeah,
bless.
that's what the veterinarians tell me when I call them. I want to be able to,
No,
take
be-
her there and maybe give her some medicine. And they said it's like giving 100-year-old woman
Yeah.
bypass. They said at night they said she shouldn't even be existing
19? Yeah.
right now. They said so, and
Oh.
she's forgetting everything. She's urinating all over the floor and
Yeah, just
and
like
now and then the veterinarian said that she's finding spaces, trying to look for some place where she could probably die.
Oh.
So I kind of want to stay with her, but I didn't want to do that and you not see me because I am such a prominent part of the class.
No. This is important. I mean, that's like your child. So it's important.
Yes, it is. And I don't have any children.
Yeah. Yes.
That's my child.
Yes.
For
No,
18 years, almost 19.
Yeah. And last, you don't know, you want to be there.
I want to be right here with her.
Yeah.
Now,
So
if she
for me-
prolongs till next Wednesday, I'm just going to come in.
Okay.
But if she, but they have a feeling that this is like her last week because it started Friday.
Oh.
Yeah, it started Friday.
Yeah.
But thank you, Professor. And I'm
Don't
gonna miss
worry.
the class because I really
I
want to do
thought
that.
of Arlene coming back. I wanted to say, Arlene, stay behind. I wanted to talk to her, how she's feeling.
Yeah, yeah. Well, I spoke to her and I gave her all the assignments that was due and everything.
Rico, can you just message her now? Tell her to come back on Zoom so I can talk to her.
Hold on, let me get my phone and tell her right now.
Thank you, Arlene. I mean,
I'll
thank
tell
you,
her right
Rico.
And so I told Michael was kind of confused. He didn't know when the exam was, but I noticed a lot of people were on it.
Right, yeah, but it's on April 5th.
Okay, I'm texting Arlene right now.
Okay.
Okay.
So she can come back on screen. I wanted to talk to her, find out. I'm praying and praying for her every day, find out how she's doing. I'm just really worried. I'm concerned that she's doing okay.
Okay, I just sent it to her.
So maybe she'll come back.
Yeah, maybe she'll come back.
Yeah, I hope she does. So we can talk,
Yeah, and my cat, she's like clinging to me. You hear her? I can't. I sit
Let
down
me
and
see
she
her. You mind?
wants to cling to me.
Can I see her, Reiko?
Yeah, come here, Kiwi.
Kiwi?
She broke my thing. So if you notice, my thing is like she sat on it one time.
Oh, hi, cute. I'm not a lover of cats, but she looks so cute.
Yeah, she lost a lot of weight.
Oh, so she was nice and plump.
Yeah, she was really plump. She was eating caviar. But she lost a lot of weight, Professor. She lost a lot of weight.
She's not, I don't know, I'm seeing her first time. She's not looking like sick to me.
Yeah.
Is she okay? Like, you will know because you're
losing her bowels.
Ah.
And urinate, and she's not, she ate a little bit today. So
Oh.
I'm grateful.
So what you're giving her, liquids or no?
And I just put out the water and her food, that's it. And hopefully she'll eat it, or she's not drinking a lot.
Yeah.
And every time she eats, she goes right to the bathroom.
Oh, yeah.
Yeah, she goes right to the bathroom, so.
And nothing can be done, yeah.
Nothing, they said they wouldn't even waste the money to do anything because of her age.
Correct. It's so expensive, right?
Yeah.
So expensive.
Yeah, they said they're not even gonna waste the money. Let me call Arlene.
Okay.
Just in case she didn't see my text.
Yes, call her.
I'm calling her right now.
Thank you.
You're welcome, Professor.
Little bit concerned.
Yeah.
About her well-being.
She's coming.
Okay, great.
She said now? I said, yeah, she said she's coming.
Yeah, because 2 o'clock, I won't be able to speak with everyone on screen.
Yeah.
Yeah. So that's why I said,
Yeah, my baby. That's my baby. And I can't stop crying, Professor.
I know. When you said somebody, I didn't want to push and ask your family member what happened.
Oh, yeah.
I said,
yeah, my only child. This is my only.
In private, I like to be very discreet and confidential.
Yeah.
My
only child.
Yeah.
She lost so much weight.
Will you get another cat after this?
I don't, my fiance, he said he doesn't want any other animals because he said this breaks his heart. He can't take it.
Yes.
And what's hurting him the most is watching me.
Yes. Be emotional.
There she goes.
Yes. Hi, Arlene.
Okay, so I'm gonna go.
Okay, Reiko.
Okay, bye-bye.
Take care of yourself and the cat.
I will. Thank you. And I'll see you next week.
Yes. Okay.
All right, Arlene. Love you.
Okay, bye.
Bye-bye.
Hi, Arlene.
Hi, Professor.
I wanted to ask you to wait behind just to know I was concerned. How are you doing?
Good, I'm doing good. I just can't lift up, nothing heavy. Not until next week I will know exactly what it was.
I am praying and praying every day in my novenas, every day in my prayers from last week when you told me, be optimistic. God is your strength.
Yes.
So don't worry, everything will work out for the best.
Yes, it will.
Okay.
Yes.
So I wanted to ask you, Aline, I thought you were the one who said you'll wait behind, but I wanted to say to you in class, Aline, please wait behind. I want to talk to you. But then sometimes, students will say I'm biased towards you.
Yeah, I know, I understand.
So I quickly asked Reiko. I said, oh, I thought Aline would stay behind. I didn't
No,
know.
I was coming back to the next class, so I would have talked to you anyhow.
I just wanted to be discreet and confidential.
Yes.
With your personal. And what's her name? Reiko asked me last week. I said, yeah, she told me about it. I prayed for her. Everything will go well. Last week, right? You were going?
Yes.
And
yes.
so then I said to Reiko, she said, can we pray? And I said, yeah, but I as a professor I cannot announce it in class. I said, Reiko, if you want, you announce it as a student to your friends in the class, that we'll just say a little prayer for her. But I don't know, Reiko probably thought, no, it's not the best idea. So I didn't want to initiate because now with the rules and regulations of everything going on, and I said, let me be careful. I don't know how students can react, other classmates.
Yeah.
I'm a professor who really cares for my students, who is sympathetic, but it can be misconstrued and misinterpreted by some. So I said, let me be, there are too many things going on, with this, with these training, sexual harassment, crime against violence, students can misinterpret what I'm saying.
Yes, that's true.
Think
Yes.
I'm being biased. I mean, you're a mature student who can understand. So
Yes.
not everyone in the class can understand. So I just refrained myself. I wanted to. And then I quickly thought, okay, Reiko, you can initiate it. If you want, I don't mind. We can have 2 minutes silence or something. So I was wanting to ask you. And then I said, Reiko, do you have? So she texted you, I think. Then she's saying, probably she won't see my text message. Let me call her.
Because I just now. Yeah, I didn't see it until just now.
Right. So that's how she called you.
Yeah.
But I expected her to be on screen, but she wasn't. So she said she's going off. Her cat is not doing well.
Yeah, she told me yesterday her cat is dying.
Yeah, so that's what she was talking to me about. She won't come next week. I said, no problem. Don't worry. And now you, Arlene, what did I say? You had to give up a lab report?
Yes, that's what I was coming back for, to find out, because Rico told me, do we have to do back?
Oh, so I gave that back to you and I gave information. So if you don't have that information, tomorrow you get the information from your group and you give me that diffusion osmosis next week. Now,
okay.
Photosynthesis, you haven't done, no? You did?
Photosynthesis.
Yes, you had done it the previous week.
Yes, I did it in class. So,
right. So
right.
that
So you want...
are you, did you do it? Well, can you give that up to me tomorrow? Or
yes,
no?
I could. Yes.
Okay. So.
Yes, I did photosynthesis.
You did it?
Yes.
Okay. So if you can finish it by tomorrow, bring it. If not, you be discreet and give me both of them the following week.
Okay. Okay.
That's why
yes.
I don't like to talk in front of everyone, even in the office, everyone will be there.
Yes.
I don't want to say these things.
Okay.
I'm taking your circumstances into consideration. That's why I'm giving you an extension.
Okay.
So But you can feel free to come in, officer, if you wish, but I didn't want to talk anything about
Okay,
it.
no problem.
So always, if you have to talk to me privately, like at the end of the class.
Okay,
Like
sure.
this.
Yes.
So that, we are discreet. And then if you're not finishing it by tomorrow, the photosynthesis, give both to me the following week.
Okay.
I'll just leave it up to you to decide.
Yes, Professor. Thank you.
What you're going to do, oh, you're welcome. What you're going to do is, if you're giving both to me, just come up discreetly in between the class and just keep it. Give it to me, I'll put it in my folder.
Okay.
But give it separately, like put it on my folder or say, Professor, these two. And so, in between the class, you come. When you're carrying out the experiment, no one's around me, just come and hand it to me. Then I'll put it separately in my folder, and then I'll grade and put up on Blackboard.
Okay, no problem. Okay.
Yeah. How are you feeling? How are you?
I'm feeling all right, just a little sore, but okay.
That's what I'm thinking. If you're sore and all, and it is inhibiting you from performing, like doing any work.
Yeah, I can't do lift anything heavy.
No.
So I can't lift, I can't sweep, I can't do anything like that.
Any chores, right? So now, tomorrow, it's okay if you don't bring your lab manual.
Okay.
Don't carry it.
Okay.
Okay, don't carry your lab manual. We can always, you can always look into somebody else's, or I can even go and bring my lab manual for you.
Okay, no problem.
Just remind me.
Yes.
Okay. Now you'll be up to it for pre-lab quiz. You can manage, to read.
Yes.
Right?
Yes.
For tomorrow's pre-lab quiz. Now, I told you missed the last week's pre-lab quiz.
Yes.
I'm going to give you that. You have to remind me.
Okay.
Okay. Now, listen to me. After class, you go straight home.
Yeah.
Why don't you follow me? You wait. Follow me and you come into my office and take the pre-lab quiz.
Okay.
Okay. Now you must remember, Ali.
Yes, Professor.
Don't forget. So you come walk with me, like you stay behind at the end of class when everyone's going. You stay behind and walk with me to my office, and you can sit there and just take it relaxed.
Okay, no problem.
The previous lab quiz.
Okay.
Okay.
All right.
Yeah.
All right. Thank you, Professor.
No problem. Anytime. So, just keep me abreast if you're missing a class or if anything, we'll work.
Okay. Thank you. Appreciate it.
You're going next week. It's not on Tuesday during the exam.
No, I'm going on Monday.
Monday. But listen to me. If they have to do anything or you're not in a position to take the exam, email me something. I will give you. I have to go in and put in a feature. But be very discreet. Don't say it to anyone. Okay.
Okay.
So if you are not in a position, email me that Monday, Professor, I am not able to do the exam. Ali, do me a favor. Give me your phone number, cell number.
610.
Arlene, I'll write it here. 610.
809.
Yeah.
4925.
Okay. Okay. Ali, take down my cell number. I don't give students, but I'm giving it to you.
Okay.
You're ready?
Yes.
612 770
Yeah.
3038.
Okay.
Feel free to call me.
Okay, sure.
Okay, I will program your number. And if you are not in a position, then I will give a date, another date for you on another day for you to do the exam.
Okay,
Okay.
okay,
So I will have to go in and change the date and add your name, and I'll do that. So
okay,
do not push yourself for Tuesday exam.
okay, no problem.
Read, but if we don't know how you're going to feel when you
Yeah.
come back on Monday.
Okay,
Okay.
yes,
So just take it easy. Everything will go well. Be optimistic and be
yes,
positive. But
yeah,
you may, like how you say, you're sore or whatever. You may, we feel down and out a little bit. Take it
yes.
easy. You may not be 100% to take your exam, but let's talk. So you can always talk to me, phone me,
Okay, Professor, thank you.
phone me.
Yes,
Say how you feel Monday. Monday, I don't teach, I'm in the house.
okay, no problem.
So you can call me anytime. I
Yeah.
will program your number so that I know. What is 610 area code?
That's Philadelphia.
Oh, you're from Philly?
No, no,
No,
I
even
just,
mine
no,
is-
I
mine
had
is-
a phone number from there.
Yeah, mine is also Minnesota. We had a family plan for Minnesota. When my brother and all were in Minnesota, brother-in-law, I mean. So I was on their family plan. And you know what? I stuck to that number. I quoted it for years now. I didn't want to change. So
Yeah, that's same thing with me, Professor.
Yeah.
I just stuck with the number, so I didn't want to change it.
I'm going to put you on my I'm going to put you in the contacts. Arlene, your last name is?
James.
James.
Yes.
Okay. So I'll write into parentheses. Okay. And so then, when you call me immediately, I know I will, now I have to write down, I think, add a group. So now I have to write you. Okay. Anything else? Now I'll have to close this and put Zoom for the office hours.
Okay, Professor, thank you. But I will come to the next Zoom
You're
class.
coming?
Yes.
You're coming for office hours?
No, well,
Oh.
you answer me on what I have to give her because I thought I had was to give maltose and bag again because we could tell me something about that, but I gave you that already.
Which one?
Maltose and bag, I gave you that already.
The maltose you gave me.
Yes.
Right.
So just the photo.
No,
finish.
so tomorrow is photosynthesis.
Yes.
But diffusion osmosis was last week, which I told them to give today. But if you don't have the data, you can get it tomorrow. And if you
Okay.
don't if you're not finished with photosynthesis, then give me diffusion and diffusion osmosis and photosynthesis next week.
Okay, no problem.
To you. Okay.
Okay. Yes.
Take
Thank you.
care. You're welcome. So I'll
Okay.
program you. So when you call me, I know it's you and I'll pick up.
Okay. Thank you,
Take care
Professor.
of yourself, okay? And
Yes.
let me talk to you. Okay.
Yes.
Bye-bye.
Thank you. Bye.
Welcome. Bye. Mhm.